Hello, everyone, and welcome to CyVerse's Focus Forum Webinar series. I'm Shelley Litton, CyVerse's Communications Coordinator. Today's webinar is the second part of our webinar series on nanopore sequence data analysis in the CyVerse Discovery Environment, presented by Jawan Song, one of CyVerse's scientific analysts, who is with the Texas Advanced Computing Center, a CyVerse team partner. The first part was presented on April 19 by Upendra Devisetti, and the video is up on CyVerse's YouTube channel. For those unfamiliar with CyVerse, we are a NSF-funded cyber infrastructure project, and this webinar series helps fulfill part of our mission, which is to train the next generation of scientists in how to use CyVerse's computational resources. Let's quickly take care of some housekeeping, and then we'll move on to the webinar. Today's presentation is roughly 30 minutes with time for Q&A at the end. Please open the Zoom chat window and type your questions there for Jawan. Materials from today's webinar, such as the presentation slides and video, will be posted on a wiki page for which we'll email you the link later today. As was mentioned at the last webinar, CyVerse is offering a training course called Foundational Open Science Skills, which is a camp-style event from June 3 through 14 here at the University of Arizona. I'll type the webpage URL in the chat window. Also, our last webinar this spring will be on May 17 on how to scan and visualize genomes with PatMatch and Circos in CyVerse VICE, to be presented by Wayne Decatur of SUNY's Upstate Medical University. Please visit CyVerse's website events page for more information and to register for Wayne's webinar. Last, we are in the process of scheduling our fall webinars, so we'll do a quick poll at the end of today's webinar for your input about topics we may cover. So without further ado, I'd like to introduce Jawan, who not only builds CyVerse tools and apps using the latest container technology, but has expertise in next-generation sequencing and RNA sequencing. Welcome, Jawan.
Thank you, Shelley, for the kind introduction. As she said, this is the second part the Nanopore Data Analysis webinar by CyVerse. Hello everyone, my name is Jawan Song, and I'm one of the scientific analysts at CyVerse and also a research associate at Texas Advanced Computing Center. So let's get started. This part of the webinar is to demonstrate a typical workflow when you have sequencing data from Oxford Nanopore Technology. I will quickly introduce you to a workflow and also demonstrate how to run the workflow in the discovery environment. And I will also talk about their outputs and other applications that can be used along the way. Here, this is an extremely simplified workflow that you can use with Nanopore data. Note that there are many other things you can do with Nanopore data, and genome assembly is the only one part of it. For the genome assembly workflow, first you will be converting the output from sequencing machine to a human readable format. This step is also known as basecalling. And using these read files, you may want to perform a de novo sequencing assembly. And at last, you may want to check the quality of your assembly. For your information, for all the applications we have on Discovery Environment, we actually have a folder of example files for you to try out the application. When you're in the Data tab in the Discovery Environment, you can simply go to this directory as highlighted in red to access these files. All right, back onto the workflow. As I said before, the first step of the workflow is called basecalling. Oxford Nanopore Technology long-read sequencing is based on the following concept. Pass a single strand of DNA through a membrane via a nanopore and apply a voltage difference across the membrane. The nucleotides present in the pore will affect the pore's electrical resistance, so the current measurement over time can indicate the sequence of DNA base passing through the pore. This electrical current signal, also known as signal, is the raw data gathered by Oxford Nanopore sequencer. Basecalling for Oxford Nanopore devices is the process of translating this raw signal into a DNA sequence. In file format terms, this is converting FAST5 file format. You may want to something that you're more familiar with, such as FASTQ or FASTA or even SAM files. FAST5 are HTF5 formats and can be viewed using list contents of FAST5 file app on Discovery Environment. Currently used base callers are Albacore, Guppy, Scrappie, which is the one that we are going to use today, and also Flappy and Triron. Albacore is the most popular base caller you may have heard of. However, we are not able to distribute due to licensing issues. Scrappie is the open source version of Albacore with most of its features. There is a recent paper that reviews these different methods on this link here. So if you're interested to find out more, please refer to this text. So as I said before, our choice of base caller today is called Scrappie. It is open source, so you can read through the script if you're interested. And it has been the first of Nanopore's base caller series that tried new approaches such as implementation of neural network. The documentation of Scrappie is available through Nanopore GitHub page. Okay, let's get started with our first demo. You can access Discovery Environment at this URL, de.cyverse.org. It has brief description about the Discovery Environment itself, and you can log in using your CyVerse account credentials. You can use the same credentials to access any of other CyVerse platforms such as Atmosphere. Now there are 3 icons to the left: data, apps, and analysis. For finding the application, you can click on apps and search for application name of your interest, in which this case is called Scrappie. Type that in there. Click on the app name, and you'll see a window open up with various fields regarding the application. And you'll see you can rename the analysis and also add comments about the analysis and even decide where to put the output files in your directory. Let's keep it default. Now there are only one input to this app, which is the FAST5 file, or a folder containing multiple FAST5 files. I have prepared an example file here called test.fast5. Click on that. Okay. And after checking everything is all right, click on launch analysis. Okay to that. And that's it. So this process of submitting a job is same as any other application on Discovery Environment. So the difference would be just the inputs and parameters across different apps. When it's launched, you'll get a notification that's shown here. And you can see the status of the job on the analysis tab. Here you can see it's submitted. And I ran this ahead of time so you can see what the results look like. It says it's completed. If you click on the app itself instead of the name, you can repeat the same analysis using the same input and parameters. Click on the analysis name, And you can see various output files. And they also contain even the log files, .err and .out, where you can see what the command was used to run this job. Click on any of the files to see the contents. Looks about right. And you can use this log file to if there's any problem with the analysis. Click on the FASTQ file, and you can see they are readable sequences indeed. If you actually click on the FAST5 file instead, and see what the contents are from editor. However, as I said before, there's an app to review this file format. All right, moving on to the next part of the workflow, which is creating a draft genome assembly. For this part, we're going to use an application called Canu, which is a fork of Celera assembler and can be used for both PacBio data and Nanopore data. As you can see in the schematic diagram, it goes through 3 steps to assemble a genome, where it first corrects to improve the accuracy of base in the reads, and the next is trimming, where Canu will trim the reads to the portion that appears to be high quality sequence, removing suspicious regions such as adapters. And finally, the assembly phase will order the reads into context and generate consensus sequences and create graphs with alternate paths. Here are, for your information, the documentation that Canu developers provide. And other assemblers for Nanopore sequencing data include Minimap, WTDBG, and NanoDJ, which are all available on CyVerse Discovery Environment. Here's a quick note about Canu on Discovery Environment. Current Canu application on CyVerse does not support multi-threaded run on our system. Therefore, if you're using a larger genome, it may not run to completion so fast. So if you're command line savvy, you can use virtual machines to help you out. Also, if you don't have your own Nanopore data, you can try out the application by downloading something from a publication. There's an app called SRA2FastQ on Discovery Environment, where you can paste in the SRA accession and get the FastQ file, and you can run Canu with this file. All right, now onto Canu demo. As before, you click on apps to get the list of applications available. Search for Canu by typing in the field. It's going to load, and you will see you will have multiple versions. In this case, always try out the later version first. And if you click on 3 dots on the right, you can get information about the tool. And this is same as before. You can rename analysis, add comments, and choose output directory. The input for Canu, you can choose either FASTA or FASTQ file. As I said, the example folder I explained before, search for app name. In this case, search for Canu, which is here, or simply type in the app name on the top. And there will be files available, and we're going to choose Oxford.FASTA because we are going to use Nanopore data, but there's also PacBio example data. Then for parameters, you need to choose what type of data this is, whether the file is from Nanopore or PacBio data, and also whether it has been corrected previously or not. The last parameter is the genome size. 1M stands for 1 million base pairs. And if it's larger than a gigabyte, you can simply write 1G for 1 gigabyte pairs. And for this case, I'll write 4.8 M because that's the size. Launch the app. And OK to that. Think about it for a second to proceed with the job submission. And let's see. You'll get a notification like before. Once the job is done, go to the Analysis tab, look for Canu. Let's go with this Canu app that's done. Click on the name. There's a lot of output files here you can see. First, let's look at .out file. You can see what the command was used. As I said before, if something goes wrong, you can check out the .out files and .er files. About right. I'll explain about the different types of outputs in the next slide. But you can see there are various FASTA files, as shown here, and also read files in gzip FASTA format. Shown here. And also, you can see GFA, like the final assembly files and GFA files. The most useful file here is probably the .report file, which has information about the run. So let's see what the report file has. It has histograms of the read length and other information. You can check out later. All right, so this is the main types of outputs that's produced by Canu. First, the .report file has statistics of reads such as length and histograms. And also they keep intermediate files of reads after correction and trimming. The three files that are most important in your Canu run is probably the config file, where config.fasta is the primary assembly file. And then unitig is the split file at alternate paths according to the assembly graph. And unassembled FASTA is the config that are not incorporated into the primary assembly. The folder also has, as I showed before, the assembly graph.gfa files. The last step of our Nanopore workflow is to check the quality of assembled reads. First step to check the quality of the assembly would be to run a simple blast run and see whether roughly is in the species criteria expected. Second would be to run a more comprehensive quality check job using tools such as QUAST. QUAST stands for Quality Assessment Tool, and the tool evaluates genome assemblies by computing various metrics. And other tools that do similar types of QC include basecalling. Okay, let's move on to our last demo. As before, go to Apps and search for QUAST in the field here. Let's see. 4.0 seems to be the latest version. Let's click on that. And you can see 3 fields for adding information of the run as before. Here. And you can check change output files. This app even has README section, giving a brief introduction of the app and also licensing information. For this application, as you can see, you can add multiple input files. So let's click on Add and look for the config FASTA file that we made from Canu run. Here to set config FASTA. We have only one input, so let's go to the next section. You can change the name of the output directory. Leave it default for now. And you can look for reference genome under this scroll down menu. Let's look for E. coli genome. E. coli data. You have it. But you can also load in your own genome FASTA file. Save the genome file here. And you can add other data, other files such as gene file or open files. And there are additional parameters, as you can see. Let's leave it default for now. Launch and run. Let's look at the analysis. The job we submitted is running. Got a completed run just under it. Click on that and see where the output files are. Click on the output folder. So there seems to be a lot of output files again in different formats. You can click on report.pdf, for example. And if you have your pop-ups enabled on your browser, it will open a new window on your browser, and you can look at this file directly from your CyVerse analysis folder. This is probably not the most interesting output because we had only one config, but you can see there are different graphs of GC content and lengths of reads. All right, so what's next after this workflow? Although we haven't not covered in this webinar, we can look into improving your genome assembly using tools such as NanoPolish or Pylon, both available through Discovery Environment. And Nanopore is of great interest to us at CyVerse. We are trying to add tools that are useful for Nanopore data analysis. If there's a tool that you want that we don't have, you can request for it using request tool form. How to do this is on this wiki page here. Also, we are excited to announce that we are working with the folks at NanoDJ to have a dedicated webinar for Nanopore sequencing data analysis using NanoDJ, and also more specific Q&A sessions with them. With that, I would like to end the webinar, and thank you everyone for listening, and thank you to National Science Foundation for their support.
All right, thank you, Jawan. It looks like we have a few questions. First up, what constitutes large genome WRT Canu assembly?
So I have run a couple of test runs for Canu because I put it on Discovery Environment. I tried the E. coli data set, which seems to run fine. Because Canu generally uses the novo assembly. People are more interested in larger eukaryotic genome. If the genome is past 1 gigabyte, I feel that it hasn't run to completion within the time frame. But I have several collaborative projects where I'm trying to improve Canu since I don't have the example data set. So if you have data sets that's larger than 1 gigabase pairs, I'm happy to collaborate.
Okay, next question. Can you clarify whether multi-threaded jobs can be run on CyVerse via command line?
So on the CyVerse command line, if you use Atmosphere, you can probably create a virtual machine and run jobs on our high performance computing resource. There's a special page for letting you know how to do that. So please check that out. I can direct you to that.
How long will it take to run Canu for a genome of 1G?
So all the jobs on our discovery environment has time limit of 48 hours. So given that it ran to completion, so it's probably less than 48 hours.
Okay, and how do you determine the genome size for the parameters with Canu?
So this is estimation. So obviously, if you don't know what you are looking into, it doesn't have to be like spot on. Probably just approximation will do. But yeah, like if you search, if you are working on a species of interest, you probably know what range of genome size is.
Okay, what if you also have an Illumina run to go along with your Nanopore run? When and how would you use that to help with correcting it?
So there are tools that combine Nanopore that enhances Nanopore sequencing data using Illumina data, just because, you know, like it has shorter read length and it has higher higher accuracy in each base pairs. So there are tools for that. I can't think of example tool right now, but if you search for a tool and you're interested, let me know.
Okay, last question. Do you know whether Unicycler is useful for eukaryotic hybrid genome assembly?
I have not heard of this particular app, so you can try, I guess.
Okay, it looks like that's all for questions. Thank you, Jawan, and thank you all for attending today's webinar. If you'll stick around for a moment, I'm going to put up the poll here. Please join us in 2 weeks for our next webinar on scanning and visualizing genomes using PatMatch and Circos in CyVerse.